I'm going to now turn things over to Joe Graves, who's on our organizing committee for Club EvMed, and he's going to introduce our speaker today. So take it away, Joe.
It is my great honor and pleasure to introduce my young colleague, Dr. Brandon Ogbunu, who finished his PhD in evolutionary biology under Dr. Paul Turner at Yale University. In some sense, Brandon is an example of contingency and the role of contingency in shaping events, in that when I was a young assistant professor in the early 90s at UC Irvine, I was called into the graduate committee office to examine the file of a potential graduate student that the graduate committee was agonizing on admitting through the program. That person happened to be Paul Turner. I took one look at the file and I said, "Are you guys kidding me?" And said, "Of course you should admit this guy, because he has a much better record than I did at my stage of career. So you'd be nuts not to admit this student." And so Paul came in, and not only was he outstanding, he was the best graduate student that they admitted that year. And as they say, the rest is history. He then went on, of course, to be the mentor for Dr. Brandon Ogbunu, who I met in person. I knew Brandon by reputation, but I met him in person. And I don't remember, I think it was at one of the evolution meetings in the late teens. And we actually took a photo with Lin Chow, who had been a mentor to me when I was a graduate student, like Brandon was at the time. So anyway, here he is. His interests are in the evolution of complex systems. He uses a variety of tools, including experimental evolution. So I'm gonna let Brandon take it away. Brandon, it's your turn.
Looks like he may be frozen. Hopefully, oh, there he is.
Okay, there you go.
Am I all right?
Good to go.
All right. Let me know if there's any problems, or if any problems arise. But I'm okay for now? Great.
Everything's perfect.
Excellent, excellent. Well, thank you for that introduction, Professor Graves. It is quite an honor and pleasure to be here during these times, and to kind of kick off the second season, I guess, or a new season of these seminars, which is very, very exciting. That's how I think about things. I chose this title, "The New Tangled Bank of Disease: From Protein Space to Injection Networks in COVID-19 Disparities." My name is Brandon Ogbunu, and as you heard, I'm an assistant professor in the Department of Ecology and Evolutionary Biology. I just made the mistake of clicking on the meeting view and looking at all the people in the room, and there's many, many luminaries here, and so I'm actually petrified right now in the best way possible. I saw Randolph Nesse here, Steve Stearns is here, and all these, and Vaughn Cooper is here, all these people who I admire, whose work I've admired through the years, I'm intimidated by. But that's what makes this very, very exciting. I think what makes this specific seminar format exciting for me is the way that it works. I love how it's described as a journal club or a conversation. It can be kind of whatever the speaker wants, from what I understand. Generally, giving no instructions is dangerous for me, because you might get a sing along. But today you're going to get a mix of an academic seminar, normal academic seminar, which I think has been the structure for a lot of them. You're going to get a bit of journal club. When I was, in my brief time as a medical student, what I was told was, when we gave rounds presentations, what you want to do is you want to create controversy. That's what I was told as a guide for how to kind of do medical rounds. So I'm going to do that. But I think more than that, I hope it's generative. The goal of these types of conversations are to generate coffees and beers, right, as I say, and in which invariably in my career become papers and grants. So that's the idea, right? We can generate interesting conversations. Hopefully we can continue to talk about these topics. Now, before doing anything, I want to thank the large number of kind of supporters of this work through the years. I think my intellectual development is a very kind of incremental process, right? Much like adaptive evolution, or more like more like genetic drift, frankly, in terms of kind of all of the contributions, and they come from random places. But I want to thank all the people through the years that contributed to the intellectual development of these years, of these ideas through the years, and I want to make sure they are properly acknowledged.
Hey, Brandon, can you hear us? We lost you.
Yeah, we okay?
Yep, you're back.
Am I back? All right. Interesting. I usually don't have problems. I don't know what's happening. So I just wanted to make sure I acknowledge all of the intellectual contributions to this work, and all the people who had who contributed through the years. Maybe we change a setting here. All right, great. All right. Thank you to all of the sponsors, all of the organizations and institutions who support this work and support this seminar. Various ones that I've attended the meeting of the International Society for the Evolution of Medicine and Public Health. It's kind of very exciting to kind of see that come together as these societies come together as an entity through the years. Again, I remember when this was a young field where there was no kind of organization, but I think it's really, really great to see this now organized into a real entity. Charles Nunn and Meredith Spence Bululu, and all of you for attending. I want to spend special time thanking Dr. Joseph Graves. I think it was great how Professor Graves was able to mention contingency, the role of contingency in our relationship. I think there's kind of another set of concepts, one that's going to be very central to what I talk about, and that is ecology. And ecology is something that I think about quite a bit, and how we can implement it into evolutionary medicine. I actually first learned of Dr. Graves when I saw him speak at the NIH. I think this is in the early 2000s, when he was talking about his book, his kind of really, really excellent book, The Emperor's New Clothes. And I saw him in the audience, and I saw this as an evolutionary biologist who was kind of doing this work that was socially relevant. It was very, very important to me as a kind of a developing scientist at the time. But more than that, Joseph Graves has paved the way. He's kind of changed this field in some very, very important ways. It's made it easier for me to do my job. So I want to make sure that's properly acknowledged. Ecology is going to be a theme, and the way the environments work, how it kind of paves the way for others and makes evolutionary, how it makes evolution possible. Okay, so we'll start with the tangled bank analogy. I think this is kind of obligatory in a lot of talks of this kind, and we've all heard this, we've all read this about kind of tangled bank. I think this is invoked in a lot of different ways when you go to an ecology meeting or an evolution meeting. Sometimes it's about often it's ecologists talking about how evolution needs to consider ecology and thinking about its problems, which is true and is going to be a theme for today's talk. But the way I think about this is kind of reframing, right, disease as its own kind of tangled bank, where like all of these, really pick any disease of any kind, it really is the product of these type of complex interactions between actors and agents. And I think this feeds kind of directly into kind of when I started teaching evolution in 2015, we did the evolution in Darwinian medicine section of the course, I had my own definition, right, which is quite similar to some of the ones that existed from going back to some of the people in the room here, understanding the evolutionary history of objects, actors, replicators, and then how environments can reveal maladaptations that cause or exacerbate. So I think that one is kind of consistent in many ways with some of the definitions that we heard. In the 5 years since then, I think I've kind of intellectually matured. I've politically matured. I think I've kind of a lot of things about me have changed. I've made more observations. I've done a lot of new and different science of my own. And now I'm at a point where I want to reconsider and redefine this field for myself. And so really what you're going to hear is me kind of really thinking through my own work, frankly, and how I can be more rigorous and vigorous and courageous with how I ask the questions that I'm asking about in light of evolutionary medicine. So really how I think about it now, right, is more like this. It's not so much replacing the other one. In some ways, it's a kind of an addendum, right? The use of evolution and ecological reason to disentangle the many actors involved in disease. The merger of modern technological, statistical, evolutionary, and evolutionary reasoning to create new biomedical metrics, heuristics, directions, and avenues. Now, disentangle is a key word here, because I think as I've continued to learn about disease, and I knew this, I've known that disease is a complex phenomenon for some time, but I think the term disentangle is one that I'm hanging on when I think about the way we can use this type of reasoning to address disease. Right. So really, how do we do the things that I just mentioned in terms of this definition? I think there's kind of four areas where I'm thinking now, and I apply this type of reasoning to the way I work now. When I think about these disease processes, when I'm walking kind of my evolutionary reasoning into these problems, or I'm thinking about the way evolution can help us understand them, I kind of consider now these four things. And I think these have not only changed my approach to doing the work, they also are kind of piqued my interest about new problems that haven't classically been a part of my research program, but perhaps might. Okay. So engaging ecology, right? How does that work? So we'll start with my research program. I have a kind of research program that kind of lives here. We do everything from, right, we start with, the way I think about the work is we can start on any corner here and move to another corner, right? We can use anti-malaria resistance to ask basic questions in protein evolution, or about drug resistance in general, or what have you. But I think really a lot of my lab has really focused on protein evolution, oftentimes proteins that are involved in disease. And kind of the analogy that I use, and one of the things that I try to do, like, Professor Graves and colleagues, is, communicating science is very important, but I think the communication of science, and certainly Vaughn Cooper does excellent things in this way as well, making it kind of tangible to people. I try to work the communication of science into the actual science. Let me give you an example. And I think one of my intellectual heroes, right, is John Maynard Smith. And of course, I wrote this reflection in genetics. That's a 50-year kind of reflection on this paper by John Maynard Smith in 1970. Many of you have seen this. And what John Maynard Smith did in this paper is, it was actually it's a fascinating back story to this paper, but basically talked about the incremental process of protein evolution, right? And so what he talked about here is that the process of moving from one end of this space to the other end of this space by replacing one letter at a time. We won't belabor this point, right? But it was kind of an effective kind of means of explaining the incremental process of evolution. It was actually a rebuttal to an individual who was at the time, we didn't really know, but was an intelligent design, a purveyor of intelligent design. But we can ask this same question. We can say, okay, well, if each of those letters are, right, we're supposed to be amino acids in a protein, well, are there actual proteins in nature, in the world as we've seen it, that actually have this exact structure, right? And the answer is yes, right? We can think about that. So resistance to antifolates in Plasmodium falciparum, is, well, not all antifolates, but to pyrimethamine in this case, is mediated by mutations at 4 sites. And so we can reframe, right? We can reframe that original analogy from 1970 that looks like this, where in this, which the path you see there is the most likely path because all of the words make sense along the way. That's how the word game works, and that's how evolution works. We can actually ask the exact same thing about this the amino acids in this resistance protein. This would be the wild type. This would be the quadruple mutant. And we can ask the exact same question. This work was pioneered by Dan Weinreich and company in 2006. Had a very profound impact on my career. But using this analogy, we can begin to think about, this is a richer analogy, maybe even than we thought, right? We can kind of think about, does evolution have, we talked about contingency, and I think this work is famous because it kind of tested the evolutionary contingency, that evolution has directions and there's things that we can conceivably predict. But more than that, I saw this as an opportunity to ask a different set of questions. And that was, about to what extent is predictable, but what forces are shaping, right, evolutionary trajectories. This is another area that Vaughn Cooper has been very, 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 has done a lot of interesting work in recently. But what are the forces that are shaping the direction that the words are moving, right? And this is an area that my research group has lived in for quite some time now. And really, right, where a lot of my work is focused on is the role of the environment in shaping protein space. Now, environment, of course, means a lot of things. I'm talking specifically about drug environment. Now, it's amazing when you look at this slide, we look at this now, unthinkable now in the COVID age, but this is my hometown. And I think when you look at this room here, the reason why I have all this here is if you give these individuals the same dosage of pyrimethamine, do you think you're going to get the same kind of active compound in the bloodstream? Of course not, right? These individuals are going to metabolize drug in a different way. And so whatever, let's just say all these individuals had Plasmodium falciparum, right, in their bloodstream, wreaking havoc and headed, on its way towards cerebral malaria. They would have a different active amount of pyrimethamine in the bloodstream, even though they all took the same dosage. The point is, right, the pathogen is going to experience kind of varying environments depending on the individual that it's in, etc. So the notion that there's going to be I asked the question of, well, given this, right, how would this actually influence the direction of evolution? Right? Like, yes, evolution might be predictable, but that predictability very well might be contingent upon us understanding something very, very specific about the drug environment. Now, I say drug environment and not only drug type. Now, there's two different drugs here, pyrimethamine and cycloguanol. I don't believe there's any chemists in the room, so I can get away with saying those are quite similar. And they are, frankly, I think, the biochemistry of them is slightly different, but they're both antifolates and presumably have the same target and they dock in the same target of dihydrofolate reductase. So the point is you have two drugs that are in the same class that are quite chemically similar. And so we can also ask, how does drug type and concentration influence the direction of evolution? If you get lost, you can just think back to the word to gene game. How would drug type and concentration change how the word is moving between word and gene, right? And so when we do that, right, we can ask questions like this. Yeah, maybe you have drug environment A, maybe drug environment B does this, maybe drug environment C does that. Maybe different drugs are going to have completely different kind of evolutionary trajectories, right? Which in some ways, is obvious, but I think it wasn't demonstrated very, very clearly. And really, this is exactly what we found. So along the top there, right, PYR is pyrimethamine, and I'm just showing kind of different drug concentrations. And the key thing is here is that you're seeing that at low pyrimethamine and intermediate, you get this the fitness peak, as we call it in the parlance of the fitness landscape. It's going to be here, and at high drug concentration, you're going to kind of go all the way to triple mutant. Interestingly enough, cycloguanol, which I told you was supposed to be similar, right, as a drug, and it's supposed to be doing the same thing, has a different solution, right? Has a different solution. I think the fitness peak at low cycloguanol is here, and at the intermediate and high concentrations, it's actually another part of the this fitness landscape, the protein space. And so we can say is the direction of evolution depends on drug type and concentration, right? Resistance management paradigm will need to be a personalized paradigm. And I think I saw Carlo Miley in the audience. There's some really, really people who are doing this work at a level that's very, 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 very sophisticated, particularly in the cancer world. And but key thing is the ability to predict is limited by the understanding of the environment. Now, I think this work was foundational for me, and I did it kind of really back in 2015 and 2016 is where this discovery came to me, before a lot of the modern kind of steering of evolution work was going on. And I think this is the thing that really, really, really hit home to me about how the role and importance of ecology in understanding, right, disease outcomes. All right, and that this dimension had been largely ignored, at least by me, perhaps all of you considered it, was something that sat with me and would inform how I thought about things moving forward. Am I stable? Am I okay?
We hear you now.
You hear me now? Okay.
Yeah.
You're back.
All right, good, I'm back. I think importantly, because my work, I've operated and talked to both ecologists and evolutionary biologists, ecologists kind of have had the norm of reaction as a very important part of their, an abstraction for quite some time. I think thinking about the way the environments change performance is a very, very central part of the way ecologists think about everything that they do. Whereas evolutionary geneticists, which is how I was trained since I was a postdoc, less so. I mean, certainly we have very good models for evolution in changing environments and things of that nature. But the notion that subtleties of environment can profoundly change everything about our population genetic estimations is something that I had taken for granted. And I think it's something I'm continuing to work into my evolutionary medicine research program. Relatedly, right, is this notion of disease as a complex system. Now, the notion of some complex system, this is a new and young science. And, you know, some people think, I think when anytime you try to introduce a new field, people roll their eyes and go, well, we don't really need a new field. But certainly this is a robust one with really smart people in it. And I think this is kind of one descriptor of what the field of complex systems looks like. And I found it to be a useful paradigm for me because of the methods that it introduced and the way it turned a lot of things of the way I thought about disease on its head. Now, remember something, I actually did have preclinical training, right? I remember what it was like, right? Being in a course and taught how to deconstruct a disease. I mean, and it's embarrassing to think about the way I was taught about diagnosis and treatment relative to what I understand now about how complex diseases are. And one thing I love about kind of the organization, get to COVID, this is obviously manifested in the context of COVID-19. But public health is a paradigm where thinking about the disease and the things that promote it as a complex system is really, really critical. And I'll give an example from work going on in my field, in my lab now. So I've been long interested in hepatitis C virus. This work actually goes back to graduate work in the Turner lab, where I got interested in thinking about indirect and environmentally transmitted viruses, which I remain interested in. This remains a very central part of my research program. Now, what's interesting about hepatitis C virus is I was interested in this. I was on the mobile AIDS clinic, you know, at the AIDS program at Yale New Haven Hospital, and so we were interested in this in light of the heroin epidemic, right? Which was like now a long time ago, you know, 20 years ago or so, 15 or 20 years ago. I mean, still is around, but that was the addiction problem we were dealing with at the time. And that's what we were considering, right? And that's when I did all my reading. Well, it's changed, right? Because now we have an opioid, right, epidemic. Quite a bit of it by individual people who inject drugs, PWIDs. You'll see that, and you saw this last year. Like the odds of dying from accidental opioid overdose surpass those of dying in cars. I mean, this is a major problem, and of course, it's been exacerbated. Right? We fear that this will go up even further with the job losses and what have you from the COVID-19 pandemic. So here we have kind of like I was interested in this problem, right, in the public health problem of how viruses are transmitted through the environment for one reason, and then kind of like unfortunately, like a new problem came that made the problem also relevant. We're actually quite afraid now, and the data are actually still, because it's a relatively young epidemic, we're still getting primary data on how hepatitis C virus is spreading in these populations, right? But nonetheless, what we did, and this actually was before COVID-19, obviously, is it's kind of amazing. I mean, I tell graduate students, write your notes very, very carefully because you might pick them up a decade later and they might be relevant for a different reason. And so I ended up digging up some of my old math models from when I was a graduate student, and they were still kind of relevant and new, and people hadn't really touched this problem 10 years later. And so what I did was we compared the way needles function in the ecosystem of a hepatitis C virus epidemic to the way mosquitoes function in an arbovirus sense, right, in a mosquito-borne disease sense, right? And it turns out there's some elegant mathematical modeling to consider, right? And now obviously they're different, right? But I think there are things about the way these mathematical models are constructed that are analogous, right? And so we published this paper in 2019 that kind of made this comparison very directly. You'll see here, modeling this individual compartment of needles kind of gives you new and detailed insight into the dynamics of disease. But I wasn't satisfied with this. I said, well, you know, the problem with the injection drug use problem, the challenge with the injection drug use problem is it's not, the assumptions of a model like this are useful to some degree, but it really is about connections. It's really about groups of individuals who are connected in these various ways. Okay, and thankfully, because of my indoctrination by the complex systems people, which largely happened when I was in Vermont, I was kind of armed with this network theory perspective, which has now become a part of my skill set. And so really, right, the disease spread in a network and right, is really about kind of who is connected to who is using with who, and what have you. And part of the reason why we call this a silent epidemic is because we don't know a lot about the individuals involved, because these, you know, we don't have great data on people who inject drugs. And it's situations like this where models can be useful, and then they are ideally and are in this case informed by the empirical results, but they kind of allow us to get a handle on how this problem might be understood and addressed, right? So that's what we did. We kind of thought about, okay, what if we thought about this problem, when we already used kind of standard mathematical models to address, what if we thought about this in the context of the way people who inject drugs are connected? And specifically, how would this inform the way we intervene, be it via treatment or via harm reduction policies, which are, you know, it's amazing. The language around harm reduction is far more progressive than it was, right, during the heroin epidemic. And now there's kind of a very open conversation about, not open enough in the United States, but certainly better than it was in the 90s and 2000s. But we can begin to kind of use network theory to think about this. So one of the things we've observed is that directed treatment is more effective, but meaning you want to identify individuals who are at the center of hubs of people who inject drugs. Okay, people who are if you're finding highly at-risk individuals in people who inject drug networks, locating them and treating them, and the drugs we have now are quite effective. Right, we see a larger decrease, and you see this is randomly selected, this is degree selected. You see a larger decrease when you start treating at 5 years, right, in the number in the infected population writ large. So if you treat individuals who are kind of at the center of populations, we can actually quantify what that would be. And on the right here, you see the equilibrium change level of disease difference between randomly selecting people that are in a city or what have you, rather than focus on individuals who are highly connected. Similarly, we can think about questions like this, right? Okay, well, what if we combined, right? Let's just say we combined randomly treating individuals who show up at a clinic and are treated. What if we combine that with setting up a shop, a mobile van, or a temporary safe injection site, so where individuals can kind of come, and this has been tremendously effective in many places as a harm reduction strategy. Not here so much; it hasn't caught on nationwide yet. But if we combine therapy, right, with if we combine therapy with safe injection sites, I think it's pretty clear that we're going to get a benefit. But I think what you're seeing here is this is the additive effect of what these two things do individually: safe injection sites and random treatment. This is actually what happens. So there's kind of a nonlinear, right, positive, epistatic effect for the evolutionary geneticists in the audience between these two intervention strategies, right? And you only can get this type of insight if you consider the problem hepatitis C virus and opioid use or heroin use as a complex system, and you invoke the tools from that field in order to better understand it. And I think this has kind of opened up another really fascinating portal of my research program, where we actually hope to now go into the field work and actually do this type of work and collaborate with kind of ethnographers, right? This is going to be a really, really important part of this. Is the social sciences are going to be key in understanding the way we address the people who inject drugs problem. And as I observe, as we discussed briefly, in the COVID world, this will be really, really important. All right. Next, we'll talk about the introduction of new metrics and new heuristics. And this is something I'm really, really interested in, right? We've long known about the problems with BMI and how it is predictive. Some people aren't. We're kind of making we have the same issues now happening with polygenic risk scores, which are predictive in some populations, the same scores are not in others. So I always think about metrics. It's like, what's the thing we're actually measuring? And can we use evolutionary reasoning to help us come up with better and smarter ways to measure things or identify things or, open up new kind of directions? And I think about this with regards to drug resistance. Now, we won't get into this too much, but these are the two drugs that you met earlier, pyrimethamine and cycloguanil. Remember how I just told you that you treat with them and you get different evolutionary solutions? I just told you that. Yet by most of the metrics we use for what cross resistance is, we would say that they function, that resistance to cycloguanol confers cross resistance to pyrimethamine by our classical definitions of what cross resistance is. So yes, it's true that on average, resistance to cycloguanol will confer you resistance to pyrimethamine, but treatment with cycloguanol will get you to a different part of a fitness landscape than pyrimethamine is. My point is, maybe evolution can give us more precise measurements and understandings of what terms like cross-resistance really mean. And I think this is kind of another important use. And I'll give a quick example in the drug case. And I think in the drugability context, and I don't look at this problem from a chemical biology sense. I think about it from an evolutionary sense, right? Can the ability of a portion of a genome to be targeted by a drug, especially by a small molecule drug. So it's like, how accessible is, say, in my case, a protein to a drug? I mean, it's a really, really important thing that we consider when we're bringing drugs to the market. But can we think about how can evolution add to this? Well, we'll just use this example. We're actually we have a data set from our lab that's original and new data, but we'll just use this kind of play data set that we found from beta-lactamase. So 10 different beta-lactam antibiotics, multiple concentrations, 16 mutants based on clinical isolates of an enzyme, beta-lactamase. And basically they're structured the way, we structured the words before. Let's just say this is WORD, say this is GNE, but it kind of doesn't matter about the direction of evolution. The point is you have 16 different alleles, all with differing levels of resistance to 10 different beta-lactam antibiotics across concentrations. And when you see here, you look at this is the growth rate. So you can just say fitness if that makes you feel better or whatever. For these 16 alleles across all of these drugs. And basically it's just a mess. I mean, as in, there's no kind of clean patterns. And I guess maybe we shouldn't have expected that. But the point is different alleles grow different. It can grow in some drugs and not others. They're resistant to some drugs and not others. And I think this is kind of a really fascinating kind of observation. So I thought this was a play data set for us to think about, all right, well, is there something useful or interesting we can pull out? What kind of patterns can we actually extract out of this that will help us think about how a certain kind of protein might be accessible to a certain drug, right? And I collaborated with a really, really, really smart guy named Rafael Guerrero, who you'll meet. And he is trained more in quantitative genetics. So we introduced advanced statistical methods that border on the machine learning line, but we won't call it that. They give us information about how drugs and proteins are interacting, right? All right. And he's there. See him. And what we found, and bear with me, is the effect sizes of drug environment interactions with individual mutations. Now let's walk through what this means. What this means is, right, for cefotaxime, right, this effect here, that's a positive effect. What that means is that drug concentration, that drug, cefotaxime, across the board, right, has this positive effect size interaction with the protein variants that carry mutation at the third site. Why don't we look at a different kind of representation of this? So these are negative effects. These are kind of negative effects between drug type and mutation types. S1 through S4 are mutations. So this variant here says that these 3 mutations and cefotaxime have this negative. It knocks down. It really, really is effective at hitting all variants of the protein that have these 3 mutations. This one is really, really, really effective at hitting all variants of the protein with these 3 mutations, and so forth and so on. There's also like positive effects, which mean that like some of these are basically what it's saying is these are the mutations that restore growth. These are the mutations that restore growth. So basically, if you have, and interesting left, right, this is the quadruple mutant. The quadruple mutant is really restorative when it's interacting with this drug here. And so using these techniques, we can distill down the interaction between individual drugs and suites of individual mutations, right, which is a level of resolution that we typically don't invoke when we're thinking about drugability, right? So means of understanding how drugs interact with targets. Why do certain drugs interact with proteins? This leads to questions in the biophysical realm. Now we can begin to engineer these mutations, look at the biophysics of the question, and really, really understand at a level of biophysical detail why it is that certain drug compounds are dancing in a particular way with this mutation and this mutation, but not this one and this one. And it kind of gives us a peek into the biophysics of the drug resistance problem. Okay. So those three represent kind of research directions of mine. The next one is one that's more speculation, one that I've been inspired to think about in light of recent events. But I think is one that I think I want to do so courageously. I want to have the courage to do, because I think it's very, very important. And I think, like the other examples, it invokes a deeper appreciation for what ecology is, and it invokes the need to think about disease processes as a complex system, right? And this is something that I think until recently I had taken for granted, right? So when we think about ethics and the way that plays a role, and we think about, these are things that typically evolutionary biologists like me run from, because I don't think that I'm trained for them, or I don't think that, or they just, or you know, yeah, because I don't think I'm trained for them, or I don't have anything smart to say. I think what I'm telling myself now is that's just being willfully ignorant, because these things play an actual role in the disease process. They are actually the things that are acting in disease. And insofar as that's the case, I have to kind of responsibly think about them. And I'll give some examples, some really fascinating medical problems that I think evolutionary medicine, right, and people who are in the field can think about carefully. Let's start here. I think this is 2019, okay? And some of you know where I'm going with this, right? So number one, the wealthiest female athlete in the world, one of the wealthiest women, like entertainers in the world, is Serena Williams. Estimated net worth, $180 million. And I think she's been this way for about a decade now. Of course, I think it was 2018 now, right? We remember this, okay? And she suffered from this really traumatic pregnancy and especially childbirth experience. Now, for a lot of us that have been reading the literature, right, we remember that, right, there's a long literature about African-American women and their, right, and low birth weight and infant mortality, right? And it's one of the most fascinating kind of biomedical puzzles in the world. I mean, frankly, I think. Because what a lot of the data say, right, so let's look at that passage for a second, is that even controlling for education and behavior in pre-existing conditions, there's this deep kind of like disparity when it comes to low birth weight and kind of infant mortality in African-American women. I think in Serena Williams's case, what's fascinating about this is, despite the fact that she's the wealthiest woman in the world, her pregnancy experience was like a girl from Compton, which is where she's from. Right? She is a poor African-American woman from Compton. Despite the fact that the wealth came later, her experience actually was a lot like her, you know, other women who were born in this community. And as you search, of course, because I haven't been trained correctly, when I see this finding, I automatically point to, well, there's got to be a genetic explanation for this. I mean, people have covered that one also. Okay, so it turns out that like immigrant women of West African descent are actually protective. It's actually protective, even though they have a higher West African, right, percentage. It's protective for a generation. So it seems like what's actually happening is the protective effect of being an immigrant and a Black woman goes away as you become more American. Right? This is a data set we've seen over and over again replicated. It's one of the great mysteries. Okay. Similarly, we can think about, right, this is kind of outlined, I don't know if you've seen this. I really recommend anybody interested in evolutionary medicine or ecology and medicine to watch this series called Unnatural Causes. It's a great documentary series about health inequities. And it actually covers the low birth weight phenomenon. I think it covers an African-American woman who's an attorney, and she kind of lives about as perfectly as you can live. And it's about as long, and has had, right, these serious problems with infant mortality rate. She had difficult times with her childbirth, rather. But what's interesting about it is, what is the city that they covered? This dawned on me recently, and I was like, wow, this is, I don't know if anybody else has talked about this, but somebody needs to. The city, I don't know if anybody's watched this, the city that they focused on as the city where there is the largest health, some of the largest disparities in the country, right, where Black women were having these really difficult time with infant mortality rate, and there was these other kind of interesting problems, was actually Louisville, Kentucky. It was Louisville, Kentucky, right? Which is also the city where Breonna Taylor was murdered, okay? So what I want to do as someone who thinks about evolutionary medicine and thinks about this, right, the way environment is crafting, there's a connection between a place where you can be, right, where you can be executed by the state with no consequences, and women are having a difficult time, right, with their infant, right, with during childbirth, and there's
unquestionably. Frankly, I look at this as a mismatch between an organism and their environment, right? That's certainly not something that's been a part of the ecological paradigm until now, but they had their turn. Here I come. And I think we have a chance now to ask another set of questions that are ecological in question and absolutely do influence disease, right? So I think what I call for myself to do, and certainly my students to do, is to be more courageous with the way we think about things, to read and engage the social sciences literature, which is what we've done until now, just punted these things to somebody else. The hippies asked that, or the social scientists asked that. No, this is ecology, and we're charged with understanding these things, right? Of course, this manifested in COVID-19, right? You've heard this dialogue play out over the course of months, right? Remember, it was exclusively a European disease, and then it kind of hit certain communities in the US very, very immediately. Of course, it didn't stop some people from being wrong right away, right? So remember this quote from Richard Dawkins, and not just from Richard Dawkins. He suspected that the reason why it was killing African Americans and Latinx individuals in the United States is because of different immune systems. And that Neanderthal genes, as Richard Dawkins suggested, are protective. But of course, you know, ironically, the opposite is probably true. The opposite is probably true, right? So if anything, right? So the point is, when we have these kind of complex disparities, we look for kind of these simple explanations because we think we're geneticists, we're smart, but we kind of failed to reveal how complex the problem really is. I would kind of call on myself and anyone who wants to work with me, who wants to work on complicated problems like why COVID is killing some people more than others, or why there's an infant mortality rate, or I don't know, why drug resistance is arising in some populations more than others, to be a complicated, complex system. And it's really going to arise from us engaging ecology, the way the environment is framing, right, the context in which disease is manifesting, which relates to us thinking about, right, the disease, both at the public health level and at the molecular level as a complex system with multiple actors, such that we can kind of introduce new things to think about, methods, and a rigorous and courageous understanding of ethics in society. And that might mean us reading the sociology literature or the political science literature, because I think it actually plays a role in the diseases that we try to understand using evolutionary reasoning. That's all. I'll now take questions.
Okay. Thank you, Brandon, for a great talk. That was fantastic. There are some questions that have popped up in the chat box, and we have about 10 minutes left in the hour in which people can raise their hand to ask questions. Meredith, how would you suggest that we proceed?
Yeah, so people can go ahead and raise their hand virtually if they'd like to ask a question, but I'll ask a question from the chat to get things started off. So we had a question from Jillian Pepper. Jillian is wondering if your approach to the evolution of drug resistance might also be applied to examining conditions favoring or reducing evolution toward greater virulence, such as based on behavior or ease of transmission.
I mean, the answer is yes, and I'd love to see that data. I mean, yeah. So I think the axis in my fitness landscape is just how well it's growing in drug. That could be anything. That could totally be anything. So that's a great idea. That's a great idea. And the environments don't have to be drugs. I think you got the gist of it. The environments don't have to be drugs. It could be anything. So I think it's thinking about the dimensionality of the problem, that it's multidimensional, and that the environments could be anything, and that the phenotypes could be anything. Just expands how we can think about kind of predictive evolution writ large. So great question and great point. And I really want to see you or someone else or whomever do that kind of work. Excellent.
Would anyone who's posted questions like to ask their question or make their comments verbally instead of just in the box? We have several people who have posted comments there.
I had one. I was curious to know if you're looking into the effect of environmental stress, which would include racism, poor diet, police interactions, all of that, on epigenetic rewiring of immune response.
Yep. So I'm not looking at that, but I think there is a burgeoning literature. I mean, you know, as this audience knows, epigenetics has its months, right? Like it has its days and months when it's like explaining everything, and then it's like not, and then it goes back and forth. But the notion of that there are There's data on the role. Stress is doing a lot of things. It's not just kind of epigenetic rewiring, right? It's increasing catecholamine levels, kind of standing catecholamine levels, which we think might have to do something with the child, the experience during pregnancy. So it's doing a lot of things. It has to do with diabetes. I think catecholamines in the bloodstream are just kind of doing a lot of funky things in the body. But certainly epigenetics is on the table as an explanation for some of the health disparities. But again, it's a complex system. And so there's going to be some diseases that are absolutely affected by epigenetic rewiring, and others probably where there's another explanation. But absolutely, that's an important area.
Great, thanks for that. We're getting a lot of comments in the chat about social determinants of health and how important that is, and how linking to societal and cultural influences is really important, especially when considering these racial effects. Mm-hmm. Did anyone have a question that they'd like to ask? You can raise your hand and we'll call on you next.
I see some good ones.
Yeah, and you can go ahead and answer questions and call on people if you'd like. You can call people out if you'd like to hear perspectives.
Large body, growing body of research by anthropologists on how discrimination becomes embodied as disease, illness. That's correct. I'm somewhat familiar, and certainly if you have things that you should point them to, post them in the thing, and I'll read them. That's me. I read. I try to, at least. Do I have comments on the bell curve by Murray and Herrnstein? Well, of course I do, but I have comments on the new book, which is worse. Called Human Diversity. I think if you want to read Charles Murray's op-ed in the Wall Street Journal, if you want to read somebody who doesn't understand the basics of how population genetics works at all. I mean, like not even a little bit. Professor Graves, do you want to ask anything? You're saying all kind of awesome things that I don't want to summarize,
Yeah,
and your voice is cooler than mine.
Yeah, I didn't want to distract from your wonderful presentation, but I will answer that last question about Herrnstein and Murray. I did them in the 90s,
yes.
So Brandon doesn't need to do that.
Yes.
And I also have several papers on that whole genre of thinking that came out last year. So I'll send references to Meredith for anybody who wants them. One of the things that I'm working on another book on this subject, on health disparity, and the epigenetic biomarker aging studies of African Americans and European Americans mm-hmm. are dramatically different, mm-hmm. such that they account for a close to 20-year difference yep. between African American males and European American males of the same chronological age. So this stuff, does get embodied, and it has real health effects.
And the telomere shortening too. The telomeres are short, or the telomere aging. Yeah, that's another marker for aging. But yeah, absolutely.
There's a number of ways.
Number of ways, and they're all older. African Americans.
Great, thanks for those perspectives. Looks like we have a question from Matt Mead. Matt, you can unmute yourself and go ahead.
Great question.
Awesome. Thank you so much for the talk. That was great. I'm just wondering, how do we prevent this siloing, or what do we do practically? Is it curriculum in grad school or undergrad, or mm-hmm. lower? Is it even just different departments? Like maybe the genetics department needs a lot yep. more sociologists in it, or
yep.
how do we prevent this?
So here's the good news. I mean, this gathering is an example, right? Some people broke down a barrier between evolution and medicine and created a society, right? And so UCLA is one of the groups. I mean, it's one of my favorite institutions in the country. They got an Institute for Society and Genetics. You know what I'm saying? It's not a game. They sit down and have a seminar where they bring in people who do, who are GWAS experts on one week, and then sociology. So I think more stuff like that is important. Next, I think, there's a lot of famous people on this call, okay? And I think it's up to them. Like, let me give you a charge, okay? Now, not from me, because I, for example, when it comes to me getting respect and tenure and all that, I have to do the old school way. I knew what I was signing up for. I got to write the grants. I got to get the papers. That's what I should be evaluated on, flat out. So I don't. But what I can do, and I can do with cahoots with the Vaughn Coopers in the room, and what I can do with cahoots with the Stephen Sterns in the room, is I can say, you know what? Maybe we need to broaden the kinds of things that are relevant work and are important for understanding. And I also be journal editors. You influence the things that hit eyeballs. So what I'm saying is, I think that's a culture change that I think in changing the reward pathway, if you will, and that can influence gradually, like this society did, the conversations that are had.
Dovetailing on what Brandon just said, I'm actually working with a number of journal editors, including the editor of BioXRiv and MedXRiv, Frontiers in Nature, on creating a new guideline for how we discuss human variation in papers published in those journals. So that's going to be coming out really soon.
Great, thank you. So we have a comment from Jillian Bentley that it's not just anthropology, but sociology and geography as well. A lot of scholars working on health inequities embedded in social structures in those fields. So that's a great comment there. We do have time for one more question. If anyone has a burning question, feel free to raise your hand now. Otherwise, I know a lot of people are probably just thinking about this, a very thought-provoking presentation.
Controversy. And like I said, I like coffee and beer, so you know how to reach me.
Looks like we have a question from Paul Watson. Paul, do you want to ask the question?
Thank you so much. I arrived late, so feel free to ignore this question. There are many, obviously, interacting mechanisms of aging. Mm-hmm. I'm wondering if there are any of these mechanisms or combinations thereof that are incompatible or contradict the evolutionary sort of ultimate level of analysis explanation for the evolution of senescence, the antagonistic pleiotropy model of senescence.
Yeah, so you know, that's obviously a debate in of itself. I mean, what the ultimate causes or rather the origins and meaning and of aging is. Let me answer it the best way that I can. I think none of the stuff that we're talking about with regards to how does stress and all of these things influence the shortening of telomeres and maybe epigenetic programming and things of nature, like Professor Graves talked about. I don't think any of those, I haven't thought about ways that they undermine any of our existing, that's actually a good question. I haven't thought about ways those undermine mechanistically any of the current conversations about the ultimate explanations for aging. I think those are just examples of kind of ecological things that can exacerbate aging. So in some ways they might support kind of some of the mechanistic bases for aging, but I haven't thought about whether or not they alter or change that in any appreciable way. No. I don't know if anybody has any aging researchers can feel free to chime in there.
I'd be happy to. I mean, basically, for anyone who's read The Emperor's New Clothes, I talk about this is just a simple case of understanding the variance in phenotype. And in the case of differential environmental stress, you're going to expect the phenotype of those experiencing that stress to be not as good as those who aren't experiencing that stress because they're in a better environment. So it in no way impacts, our understanding of the evolutionary theory of senescence. It just says that these individuals are living in drastically different environments. And, to paraphrase one of the comments from the Democratic Party convention, where a woman said that there are two environments in America, the one that Donald Trump lives in and the one that her father died in.
Mm-hmm.
Thank you.
Okay, everyone. Well, on that note, we are at the top of the hour. We're going to have to close out. Brandon, I want to thank you again for a great presentation. And, I'll just say to you and to everyone who's still on, you talked a lot about communication across disciplines and how much you enjoy that aspect of doing science. But one thing that really didn't come out in this conversation today is all of the work you've done on communicating with the public as well. And I'm just so impressed by all the effort you put into that in Wired Magazine, Scientific America, Undark. I've read some of those pieces and didn't, connect it with you until actually today when I was looking over your CV. That's a tremendous amount of work. I counted something like 12 or 14 articles just in 2020. And so thank you for that and for communicating these perspectives and so many other perspectives with the public. I think that's really impressive, and I'd love to know how you find the time to do that. Anyhow, thank you for a great talk. I want to just quickly share my screen and show you the tremendously exciting set of talks we have coming, if I can make this work, in Club EvMed. We've got most of the fall already set up, almost I think all the way through December. We have Mayra Eplin and Julia Butt next week talking about Helicobacter pylori and virulence factors. We have a conversation with Amy Boddy and a paper she recently published in Evolutionary Medicine and Public Health that's coming up. I saw Amy on earlier. I hope she's still around. We have Lou Muglia and some of his colleagues, Antonis Rokas and Tony Capra, talking about the evolution of human birth timing. We have Dan Lieberman talking about his new book, which is going to be published in early September. We have a student spotlight episode. We have Joe Graves coming up. We have Eleanor Carlson talking about some of her work on SARS-CoV-2, and the list goes on. And so we're really excited about this lineup. We're still, finalizing some of the pieces here, but please tune in. Please join our mailing list if you're not already on it. And we really look forward to seeing you approximately once a week. Okay, so stay tuned. Okay, thank you, everyone. Thank you, Meredith. Thank you, Joe. Thank you, Brandon. And thank you all for tuning in.
Thank you
Great to
all.
see so many people.